talk about maximizing your success in multi-parameter flow cytometry. So for some of you that might be new to flow cytometry, you might be wondering what kind of things you can do with this technology. I'm going to go over kind of like 4 different topics and what you need to get good data from your flow cytometry experiments. So the first thing is to define your biological question, right? Every experiment's going to be different. Every objective's going to be different. Next is to know your instrument. So I'm going to go over some of the configurations of your instrument, how you can read the different channels, and what fluorophores you can use at this particular instrument. Then show you how to match the fluorochromes to your antigens when you design the panel. And then lastly, the important controls and for your experiment to be able to not only get the most out of your data, but to make sure that you get consistent data here. Okay, so flow cytometry, you can do a lot with this technology. So it can be as simple as doing multi-parameter surface phenotyping to intercellular protein detection like interleukins or cytokines to functional studies. And then here's just some quick like visual graphs of what each of those are. So this is just your basic immunophenotyping. Some of the assays you can do can include proliferation using our cell trace violet dyes amongst other nuclear dyes. If you're looking for viability to compare live and dead cells, you can use our fixable viability dyes or our live dead. You can study cell cycle by Chi67. Cytokine, interstellar cytokine like interleukins or interferon gamma, and phosphoflow. And so that's not all. You can do a number of these things, right? So there's really is a large amount of applications you can do with flow cytometry. Pretty much if it's fluorescent, you could probably run it on this instrument. And I'd be happy to discuss, any of these applications. And in fact, in the next series, based on whatever feedback we get from here, we can tailor the next presentation to kind of be applicable to any of those applications that you may be interested in. And so here's just to drive that message home, how important this flow cytometry has been over the course of the last 2 decades. So this is just publications that have reported flow cytometry graphs or images, so data. It's a little old, so the last one was 2012, but you can see that it's steadily increased, and it has since that time. And so here with Thermo Fisher, we're uniquely positioned to address all your flow cytometry needs. So you are familiar now with the Attune NxT, which is the cytometry flow cytometer. But we can also support you with the conjugated flow antibodies. There is RNA flow. So not only can you do your standard protein flow cytometry, but you can also look at up to 4 mRNAs at the same time. Cell health, where you're looking at apoptosis, viability, or to say mitochondrial health. And then your everyday essentials like beads, buffers, and controls. And so just a quick slide to kind of introduce my colleague, Ashley Manderville. So she is your main point of contact for all life science needs. And then I support the flow antibodies and reagents. And then our colleague Russ Tong, who's not here, he's actually the Attune specialist. Okay, so the first step, define your biological question. So that we kind of went over that with the different applications, right? So I can't really go over exactly what each and every one of you does, but having in mind, having seen those different applications, you can use this instrument to look at those applications further. And so here's getting to know your instrument. You guys have the Attune V6. The reason it's called the V6 is because off of the violet laser, you have 6 channels. You also have the blue laser, the yellow laser, and the red laser. And this graph, and also in the little pamphlet, you should have like a fluorophore selection guide, which would include pretty much all of our fluorophores, dyes, and reagents that could be detected from each of those channels. I'm going to highlight quickly the violet laser because there are 6 channels. So it's your laser that has the most channels available. So it's a lot of real estate here. So for that, we have the super bright fluorophores, which are these polymer dyes. The base is actually the super bright 436, and then the super bright 436 polymer in tandem with a bunch of other different acceptors that are also fluorophores make up the rest of the family of super brights. That's 600, 645, 702, and 780. So these dyes are actually really, really bright. They've been validated for flow cytometry. They're very similar to the brilliant violets. So some of you are familiar with brilliant violets. They're also polymer dyes. They can expand the number of dyes available for the violet laser. So as I mentioned, you guys had the Attune NxT has 6 channels available off of the violet laser. Here are 5 different violet fluorophores. So you could use 5 of those 6 channels just on these fluorophores, right? And so there's space here in this part where you can add a viability dye or another fluorophore that we have like E4506 to extend the full range of the system. And so in this graph, really the highlight here is on the y-axis you have, well, actually I should go back. Let me step back. For some of you that haven't seen a flow plot, this is called a dot plot. So this is how a lot of the flow data will be presented as. Each dot represents an individual cell. And so on the x-axis here, we have CD20 tagged with PE. And on the y-axis, you have CD11C tagged with super bright 702. And then here's PE and APC. And really just the point here is to show how bright super bright 702 is. You see that the negative population here and the positive population here is there. And you see a greater separation with the super bright compared to APC. And APC is truly a bright fluorophore as well. And then later on, we'll talk about how to maximize and continue to differentiate the negative and the positive population using culture control. I'll just skip this one here. Okay, so this slide's here to kind of validate our super bright dyes. They are tandem dyes, as I mentioned. And so oftentimes there's a fear of tandem dyes becoming unstable after fixation or losing some of their brightness. And so in red, we have fresh cells, so unfixed, just stained and then run on the cytometer. In blue are 30 minutes fixate after fixation, orange a day, and in green 3 days. And you can see, so again, they look at the x-axis, the separation. So the higher the cells are on the right, the brighter they are, or the more CD3 the cells have, right? And so you don't see a change in signal intensity across those times. So these dyes are really stable, and you can store them in fixative for up to 3 days. Okay, so now that we've covered some of these reagents that you have available to you, we can talk about matching your fluorochromes and antigens. And so this really goes into kind of the theory or background on how to design a flow cytometry panel. So there's 3 different groups you may want to consider when designing panels. When you think of your primary markers, these are your well-defined, very almost like lineage heavy markers. They're broad subsets. They'll either be on or off. So you'll either have positive signal or negative signal. There's not going to be an in-between or a spectrum. And an example of this would be like CD3 and CD4 on T cells. Your secondary are also well-defined. The markers should be relatively bright or expressed highly, relatively. But they may be in a continuum, right? So you may have cells that are low, intermediate, and high in expression. An example of that is HLA-DR. And then your third group is your tertiary. This is probably where you're most interested in, the markers that define your more interesting population. It tends to be low expression, or maybe you don't know, so it's uncharacterized. Maybe this is the first time someone's looked at it, and so you're unsure if it's going to be expressed in your system high or low. So in this example, it'd be CD25 or some unknown, right? And so what you want to do with each group is you want to apply these rules. So one of the first rules is, reserve bright channels for dim or unknown targets. So this would be your tertiary group. So bright fluorophores or bright channels would be like your APC, your PE, and your super brights. Next point to remember is that mutually exclusive markers should, you should have mutually exclusive markers on overlapping channels. So when you have, say, a V1 or a V2 channel next to each other, you don't want to put, you want to put mutually exclusive markers on those two channels. You don't need to have space in between them because the same cell won't express that marker. And the converse applies too. So for co-express markers, you want to make sure that you have those spread out across the spectrum. So you wouldn't want to put something like on V1, like CD4, and then on V2. Going to cause some issues later down the road. And then you want to minimize spillover, particularly in these channels, APC E4780 spilling over to APC, and P-Sci5 spilling over to APC. A good indicator sometimes, you can use a spectral viewer. We have a web page where you can kind of visualize all the fluorophores, but you can find a number of web pages to do that. Also paying attention to the filter channels across the different lasers. If you see that a filter channel is very similar or exactly the same in one laser versus the other, you might want to be careful in making sure that those two channels, you put mutually exclusive markers. Okay, so here's, I just put this slide again to kind of hit the, so you can visualize. So for example, if you're doing something on VL1 and VL2, you wouldn't want to put markers that are co-expressed around those, right? Because they're too close. You can easily do something like FOXP3 here and then CD4 here instead of putting them right together. And then with that same thing, you could do here CD3 and then CD19 because they're not going to be expressed on the same cell. Okay, so kind of for these tertiary ones, this is the first thing that you're going to want to when you're designing the panel. Those are the ones that you're going to focus on first, because you want to make sure that these dim markers or these unknown markers get the best fluorophores. So you're going to put them on bright fluorophores like PE, APC, or some of the super brights. You're going to want to use like your medium or pretty bright fluorophores. Again, super bright 600 and 645 are bright, but they're not as bright as super bright 436. And then lastly, your dim markers and your very well-defined subsets. You could use your dim fluorophores like FITC or APC Sci7. Okay, so then the last step, which is probably a little bit probably more complex step, is setting up the instrument and having the appropriate controls. So one of the first things you want to do when you're designing, when you're getting a new antibody, is titrate it. And here I'm just going to show you stain index. I don't know if anybody's familiar with that. That's really just kind of gives you a broad idea of how bright a fluorophore is in your system. So like I say that PE is brighter than FITC, and the way I do that is if I stain, say a cell with that's CD3 with FITC, I can use that same cell and stain it with CD3 PE, and then follow this formula and take the mean positive minus the mean negative over 2 times the standard deviation of the negative, and you'll get a number. And so you'll see a difference between the FITC and the P. The P should be higher. It should have a higher stain index. And so when you titrate your antibody, say this is the recommended concentration that the vendor, like us, we show. You can do a titration, go look maybe like a 5-point titration, and then plot it on a graph and pick the range that's more optimal right before it starts to decrease, right? Because as you dilute your antibody, you are going to see less and less signal. But there is a point where you can get a sweet spot where you're not seeing that decrease, and you're actually seeing an increase in stain index. Similarly, the instrument also has a sweet spot in terms of voltages. So a higher PMT, so you turning up the voltage, and this is probably more familiar if you're used to running a flow cytometry experiment. as you increase the voltage on the instrument, the staining, the positive population will shift to the right, or will shift up, right? So you'll see more staining. So you can control the population, how bright you want to see it, just based on the voltage that you use. But just because you increase the voltage doesn't mean that you're going to enhance the resolution. Similarly, if you have too low of a PMT, you might not be able to resolve the two populations, the negative from the positive or the dim populations. And I'll show you what that looks like here. So what you want to do with your instrument is do a voltage block or voltage optimization. Now, this is kind of a daunting task if you're not using the instrument a lot, but at least you kind of get an idea of why this is important. So here is, say, a sample, just in some PBNCs stained with CD3, okay, FITC. If you put the voltage at 250, you have these populations, the negative population and the positive population. As you increase the voltage, the positive population shows higher, right? So the staining index is increased. There's a bigger separation between the negative and positive. And if you keep doing that, you also notice that it keeps doing it, right? So at 350 is higher than 300, 375 is higher than 350, and so forth. But what you'll notice is, although the positive population is going up as you increase the voltage, you're actually getting the negative population kind of spreading, and it's also going up. So again, you want to kind of hit the sweet spot where you have the greatest separation between the positive and negative. So it's not always just cranking up the voltage, because you will start seeing the spreading here. So in this example, the sweet spot would be like 375 or somewhere around there. And you can similarly graph these two values here and kind of visualize that and get the sweet spot. Now, you might not always do this when you use the instrument the first time, but at least you can visualize when you're running your control, your single color control, kind of change the voltages and see how the different populations, how the negative and the positive population travel when you change those voltages. Okay, so I mentioned spillover and I've mentioned compensation. So here's just an illustration of what exactly I mean when I say spillover. So here you have two fluorophores, PerCP Slide 5 and APC Alexa Fluor 750. And then there's two channels, the 69540 and the 78050. So this shaded box here is the 78050. This shaded box here is the 69540. Okay, those correspond to a specific channel and on the Attune or on the flow cytometer. And then, so here are the emission curves of these two dyes. And you'll notice that most of the dye or the emission from PerCP Slide 5 is covered under this channel, which is under this curve. And most of the APC Alexa Fluor 750 is captured by that filter, right? But you also see that some of this fluorescent signal from the PerCP Slide 5 is spilling over into this channel, right? You see this little bit here that's spilling over. So that's what you'll need to compensate out, this spillover here. And so I'll illustrate that with some data. So here's PerCP Slide 5.5, just PerCP Slide 5.5. There is no APC Alexa Fluor 750. So I said there's only PerCP, so you should have a negative population. There's no APC Slide 7 on this, just PerCP. But you see that it's showing as if there's some signal, right? So here's your single stain control showing you that. This is why you need to compensate, because what you're seeing here, that difference over to the other detector. So when you compensate it, oh yeah, so here's showing you the MFI, so the mean fluorescence intensity for APC Slide 7. So it should be, close to zero, kind of close to this population. But in fact, you'll see it's 1277. So it's huge. It's a huge increase. So then when you compensate it, you should have the median or the mean a lot similar from the unstained to or from your negative population to the positive population. So here again, there's only PerCP Slide 5. And you see that this looks a lot better. There's less of the APC. And then when you look at the mean or MFI, 58.3 for APC Slide 7 for the negative, and for the positive is 59.1. Okay, so that's what you want to do. And this is why it's important, because if you have a bunch of other colors, you can imagine that just compounds the problem. You have all kinds of spillover across your panel. And then, okay, so here again, CD11B, CD11C on the axes have uncompensated data. And when you have uncompensated data, you tend to see a slant. So I'll note it here and some of this here. Then when you compensate it, you can clearly see the different populations. So here's one population, here's another population, and then here's another population. It's much easier to see those with properly compensated data. So there's multiple ways of compensating. You can use your cells and do single stain controls using actual cells, or you can use beads. So there's pros and cons, right? Like one of the caveats of using cells is if you have limited cells. So if these cells are precious, you may not want to use them up on just controls. Another time you may not want to use cells is if the marker or antigen is dimly expressed, or just the number of cells that express that marker is low. Because when you do your compensation, you want to have a good amount of negative cells and positive cells to then run as your control. And if it's too dim and you can't really tell where's the positive and where's the negative, or if there's too few, you might not get good compensation. Beads are great because you have excess numbers, so you don't have to worry about running out of beads. You have millions of beads. They're very bright. They will always stain any fluorophore bright. You don't have to worry about dim markers because they will capture the antibody regardless. And you have a nice distribution between negative and positive populations. So it should be 50-50 when you use beads. And I'll show you here an example. So this is if you had an IL-13 PE and Percy-PE 4710 data, and you had to guess, or you had to use cells for compensation. So how would you know where your positive cells are here to designate those, right? It's a continuum. It's hard to make a gate. And even if you did have some, it would be very few compared to the negative. Have beads, they're roughly the same population or same size population. It's very discrete distribution. You have a negative and you have a positive. Very easy to identify the positive population. So this is why it's recommended generally to use beads. Okay, your fluorescent -1. So fluorescent -1 is another control. This would contain all of the markers except the one of interest. So that's why it's called fluorescent -1. So if you have a 5-color panel, this would contain 4 colors, and then your one of interest. And you do this for each one of your markers. It helps control for the spread after compensation. And I kind of mentioned that earlier. As you compensate, you see that the population showed, oops, if I can go back. Actually, I'll go forward and show you that. It controls for the spread after compensation. It's important for low density or smeared populations because it helps you identify where to put the gate, whether it's the negative population or positive population, because you can't use isotype controls or unstained. And again, so it allows delineation of positive from negative. And so here's an example. So this is CD8 PE size 7 with on the x-axis, and then on the y is TIGIT P. If you were just using just like a negative population or positive population, it looks like the majority of the concentrated here. So this is a density plot that shows you the contours. So based just with your eyes, you would think that this would be a good line to separate the positive and the negative, right? Because the majority of the population here, and then you have a smear on that side. With FMOs, though, you can see that if you put everything but the PE, you see that there's a little bit actually going past that line on this one. And you see a little bit going up there too. So using these FMOs, you might want to draw the line up there. So now the better point to put these quadrants would be to lift it up to that red line. Right, so this is why it's important, because you're getting some of that spread from compensation. And so here now you're adjusting that and making sure you're putting the gates in the right place. And like I mentioned, you may not all controls are made equal. You might not want to use an isotype or an unstained. And here's why. So these are interferon gamma PE stained cells, just autofluorescence on lymphocytes. So this is a histogram. So it's just instead of a dot. In red is the FMO, which is gated on the subset that you're interested in. In green is the mouse IgG PE isotype. And you see that it's already shifted more than the FMO. Unstimulated cells would be like a biological control. You see that histogram. And then once you stimulate the cells, it actually shifts. The whole autofluorescence shifts greatly. So which one do you use as a control, right? You have all these different ones, but depending on which one you use, you'll get different gating strategies, or gate, sorry. So in this situation, internal negative might work best. So here, say these are stimulated cells. You can see that this population here is not the one of interest. So you're interested in the CD3 positive interferon gamma positive cells, which are these. You can use this population, which is non-CD3, to draw your gate. So you just do something like that. Okay, so there's multiple controls that you can use. And you kind of have to choose what makes the most sense for each application, for each occurrence. I think this might be the last point. For analysis, you want to exclude the doublets, debris, and dead cells, and I'll show you why. So here is the forest scatter of our population of interest. When you then look at CD4 and CD19 from all of these cells here, this is what the plot looks like. You see that there's a population here, here, and here. But then there's a lot of stuff going on in between, right? So to clean that up a bit, because it'll help you kind of visualize and separate these two populations. Instead of just going from forward scatter to there, you can take the forward scatter, gate on that population. Then from that population, gate on singlets. So this would be cells that are not paired with any other cell. They're not adherent, stuck, or for whatever reason, they're singlets in the suspension. So you gate on these, remove all of this, and so that cleans up your data like so. But you still have some stuff there, right? So the next step would be to use a viability dye. So then using a viability dye, go back to your forward scatter. And in this case, it's fixable viability dye E4780, which is an amine reactive dye. So the positive cells are actually your dead cells, and then the negative cells are your live cells. So you gate on the negative cells here, and it cleaned up the data so much more. You can clearly see that there's one population, there's another population, and another one. And you see that you can better define the two populations that are next to each other. Okay, so it's really important to keep in mind to always use, or to always exclude the doublets, debris, and dead cells. And the reason why you kind of remove dead cells is, one, you don't really care about looking at your stuff from dead cells, right? That's not what you're studying. But two, dead cells can be rather sticky. And so they might take up antibody that wasn't intended. So something that just bound just because it was sticky. So I think, okay, so yeah, so I have a slide on our live dead fixable dyes. We actually added quite a bit more. So even then with this, we had it for every laser, but now I think we added like 6 more dyes. So along with all the fluorophores that you'll need for all your markers and panels, you no longer have to worry about trying to fit in a viability dye. You know, you don't have to stick with one filter or another channel. You have pretty much all channels available now. Just kind of maximizes your panel and you get more creative. And okay, so those are the 4 steps. So first, pick your analysis, define your objective, what application you're going to study with flow cytometry. There is a ton. Know your instrument, so that might be that the fluorophore selection guide might come in handy. It also includes the filters for all those channels on the Attune. match the fluorochromes and antigens. So that would be matching the high express markers with a dim fluorophores and vice versa. And keeping in mind also the spillover. And then lastly, use the right controls. So ensuring that you have the right compensation controls, ensuring that you titrate your antibody, do your FMO controls, and make sure you have the appropriate voltage, like I mentioned, because it can really change how you can discern the unstained or the negative population from the positive.